Robert. I'm a professor for machine learning and digital health at the Hasso-Plattner Institute. Welcome to the second session on reinforcement learning in neuroscience. And we'll start out with spotlights. And Serkan Arik will do the first spotlight.
Hi, this works, right? Hi, I'm Kainan from Baidu Research. Today I'm going to present our work, neural voice cloning with a few samples. Speech synthesis is also known as text-to-speech, or TTS. When the TTS models are conditioned on both linguistic information and speaker information, we can call them multi-speaker TTS models, as shown in this figure. The multi-speaker TTS models can synthesize the voices of thousands of speakers. However, the speakers have to be in the training dataset. The multi-speaker TTS models cannot handle any new speakers. And also, empirically, they require 10 to 20 minute audio per speaker for training, as shown in DeepVoice 2. To address these limitations, here comes the voice cloning. Voice cloning is to synthesize the voices of new speakers from only a few speech samples. It's a highly desired feature for personalized speech interface, content creation, and so on. Voice cloning is a challenging task because in many cases, we can use only a few second audio to clone the voice of a new speaker. And also, we prefer to do voice cloning with low latency and a small footprint. In our work, we present two approaches for neural voice cloning: speaker adaptation and speaker encoding. Speaker adaptation is to use a few text and audio pairs to fine-tune a pre-trained multi-speaker TTS model for a new speaker. There are two options for the fine-tuning. You can fine-tune the whole model, which means both the speaker embedding and the multi-speaker TTS model, or you can fine-tune the speaker embedding only. From this table, we can see the whole model adaptation. When a new speaker comes, the cloning time for the whole model adaptation is much less than the embedding-only adaptation. However, the whole model approach is really easy to overfit, and it requires millions of parameters per speaker because we need to store the whole model for every new speaker. So in our work, we proposed another approach, speaker encoding. Speaker encoding is to directly predict a new speaker embedding for a multi-speaker TTS model. So we need to train a separate speaker encoder with audio and speaker embedding pairs. So no transcripts are needed in the training process of speaker encoder. It's text independent. So now when a new speaker comes, we will first predict the speaker embedding from only a few speech samples using the speaker encoder, and then we feed the speaker embedding into the multi-speaker TTS models. So the cloning time is just a few seconds. And all of the new speakers can share the same speaker encoder, which makes it more favorable for low-resource deployment. Here are some subjective evaluation results, the mean opinion score. Mean opinion score is to evaluate the naturalness of the synthesized audio and the similarity with the original new speaker. As we can see here, the whole model adaptation works better than other approaches. However, all of the results here are using the classical Griffin-Lim algorithm as vocoder. We can further improve the results by using state-of-the-art neural vocoder, such as WaveNet. Also, in our work, we found the simple algebraic operations to the predicted speaker embeddings are highly efficient in transforming the speaker characteristics. For example, we can use a speaker embedding of British male add the average of the female minus the average male to get the voice of British female. And the same thing for the accent. which is an interesting observation. Cool, that's it. Thanks for listening, and welcome to our poster and listen to audio samples.
Hello? The next spotlight is Sang-Woo Lee, answering questioner's mind, information theoretic approach to goal-oriented visual dialogue. Thank you.
Yeah, thank you. Hi, I'm Sang-Woo Lee at Global AI Research Neighbor. This study is co-work with Yu Jung-ho and Byung-Tak Jang and Seoul National University, South Korea. Today I'll talk about our paper answering questioner's mind, information theoretic approach to goal-oriented visual dialogue. Our main task is guess what? It is a goal-oriented visual dialogue task. In this task, there are two players. One is the questioner, and the other one is answerer. The goal of the game is to figure out the target object, which is highlighted by green mask in this figure. The answerer knows what the true object is, but the questioner doesn't know. So to figure out the target object, the questioner asks a series of questions to the answerer. The answerer can respond but by yes or no only. If the questioner figures out the target object, the win of the game is achieved. To solve this task, many researchers try neural network-based approach. So they made neural network-based questioner and answerer, and they make this agent to talk to each other. They use supervised learning method to train their agent from training data, or employ reinforcement learning to talk to each other and get a reward from the result of the game. Especially the questioner consists of two recurrent neural networks. One is question generator and the other one is guesser. However, this method struggled to find an efficient RNN model through backpropagation, owing to the complexity of a series of sentences of dialogue. So alternatively, we propose an answer in questioner mind framework, or AQM. It is a novel goal-oriented visual dialogue framework. The AQM is a method to make a questioner, not an answerer. AQM's questioner treats this game as 20-question game. It means that our system first discovers candidate object by YOLO, and then make an efficient question which sprites out candidate object in the image. For example, is this machinery is a good question because it separates candidate object by harp in this example figure. In general, we think this goal-oriented visual dialogue problem is finding a question having a maximum information gain, which measures how much the question decreases the uncertainty of the candidate object. Meanwhile, it is noticeable that there is no question generator RNN or guess RNN in AQM. Alternatively, AQM's questioner has approximate answer generator, a mental model of answerer. Using this mental model, we can calculate the information gain for question generation and posterior distribution for guessing. So it is the result. Our AQM makes tangible performance on guesswork gain. AQM achieved an accuracy of 63% in 3 turns and 79% in 10th turn, outperforming deep supervised learning and reinforcement learning method. And this is the generated dialogue. And this is the conclusion and argument. So see you at the poster session Tuesday afternoon or visual workshop Friday for our future work of AQM. Thank you.
The next spotlight is presented by Kejin Yi on disentangling reasoning from vision and language understanding.
Okay, good afternoon. I'm Kejin Yi from Harvard. Today I'm going to present to you our work on neurosymbolic visual question answering. This work is done at MIT jointly with collaborators from IBM and DeepMind. In this work, we study the task of visual reasoning. The basic setup is shown in the example here, where the machine is presented with an image and asked a question in natural language about its content. Let's first look at how a human might perform this task. When the human reads a question, are there an equal number of large things and metal spheres? The human will first try to identify all the large objects and count the numbers, and then perform the same operation for the metal spheres. Finally, he will compare the two numbers he counted and generate an answer to the question. From this example, we can see that the human reasoning process can be separated into 3 disentangled components, including visual perception, question understanding, and logic reasoning. Recently, a lot of datasets have been proposed to study how a machine might perform this kind of task. The example I have shown you comes from a dataset called Clever that was proposed by Johnson et al. Last year. The Clever dataset comes with synthetic images of objects with compositional attributes, including color, material, shape, and size. And the questions in Clever are paired with functional programs that represent the underlying logic of the questions. In order to solve a task in Clever, the model should be able to unravel the logic steps that underlies the input question and perform them correctly to the compositional object attributes extracted from the scene. In this work, we introduce the neurosymbolic visual question answering model. Our model is motivated by the intuition drawn from the previous example of human reasoning and is consisted of 3 disentangled components, including a neural scene parser for visual perception, a neural question parser for question understanding, and a symbolic program executor for logic reasoning. Given an input image, our model perceives it with a neural scene parser. The parser first detects all the objects from the scene and outputs their masks, and then extracts the single object attributes from the proposed segments, including the object's size, shape, material, color, and location in 3D. These attributes extracted from all the objects in the image form an abstract representation of the scene and serve as the knowledge base for our reasoning task. On the other hand, to understand the input question, we use a semantic neural-based question parser to turn it into its functional program. And finally, our model performs logic reasoning via a symbolic program executor, which explicitly runs the logic of the program on the abstract scene representation extracted from the image. In this example, the scene representation is sent to the first module of the program, filter cylinder, which returns all the cylinders from the scene. And so the output from the first module is sent to the second module, which will return another list of filtered objects. And this process will continue until the executor hits the final module of the question of the program and outputs an answer to the question. Our experiments has suggested that the symbolic space is a more natural venue for logic reasoning than neural net features, especially for questions with long program traces. As a result, our model is able to achieve high accuracy on the Clever dataset and outperform other state-of-the-art neural-based models. Furthermore, since question understanding is learned as a disentangled component, our model actually requires fewer questions and answers for training. Our model can achieve more than 90% accuracy when trained with only 1% of the questions, and 99.7% accuracy when trained on 10% of the questions, which significantly outperforms the strongest baseline under these 2 conditions. Finally, symbolic program execution also offers transparency and interpretability. Compared to another baseline model called IEP, which relies on neural program execution, our model is able to obtain the correct answer while accurately predicting the underlying program of the input question. Quantitative results also show a significant gain in performance of program accuracy. So as a summary, we propose a disentangled model for visual reasoning that combines neural network as powerful pattern recognition tools and symbolic program execution for reasoning. We hope that the combination of deep learning and symbolic approaches can inspire future research in various directions, in visual reasoning as well as other directions. Thank you very much.
The next spotlight is learning to optimize tensor programs, presented by Tanji Cheng.
Okay, good afternoon, everyone. Today I'm going to tell you about how we can use machine learning to revolutionize how do we build machine learning systems. So as we all know that machine learning has become so ubiquitous, they want to deploy it to all the hardware backends they want that's available today. This will include your data center CPUs, GPUs, your mobile phones, as well as like TPU-like accelerators. The existing approach on doing this actually is pretty engineering intensive. So to take a look at existing approaches, usually deep learning system like TensorFlow, PyTorch will represent the high-level machine learning computation as data flow graphs, where each of the operator node in here represents an operator such as like convolution 2D or matrix multiplication. To offload them to hardwares like NVIDIA GPUs, what we currently do is we will offload them to existing libraries like CUDN, which is heavily manually optimized. So what's the drawback of this approach? For each of the operator in the high-level computational graphs, the problem is that they have to exist in a low-level library like CUDN. That actually translates to 3 engineers, actually, as a matter of fact, more than 3 engineers who have to implement them on those low-level libraries. As you know, that machine learning is evolving so fast that either due to system optimization or like algorithm optimization, we might introduce new operators that may not exist in the existing libraries. What should we do now? One of the simple approaches, you can hire another engineer to implement that, but that usually results in a higher turnaround time. If you multiply that by the amount of hardware backend you want to support, you can imagine it's a very engineering-intensive task that you want to support. We want to revolutionize this with an automated approach to replace the human effort with a machine learning-based program optimizer that automatically learns to generate deep learning programs on the fly for different kind of hardware. In a nutshell, what we really want is that we want to build a learning algorithm that searches over billions of possible program optimizations for a given compute description. And at a high level, our learning-based program optimizer will propose programs to a target hardware, collect them as training data, use them to learn statistical cost models, and use the statistical cost model to feed back to the program optimizer to let us find better programs faster. This is a similar problem setting like hyperparameter optimization and other settings, but it does have a very unique problem characteristics in the sense that we have a relatively low cost, which means that we can collect a lot of data, but the learning algorithm have to be fast. We also take benefit of rich domain-specific problem structure. To take benefit of the high-level programming domain-specific structure, what we do is we will lower the programs to low-level abstract syntax tree, which is shared across multiple tasks, and we'll extract statistical features on top of them, build a gradient boosting tree-based model that's more like traditional-based pipeline. We also have a tree GRU-based model that's more like a neuron-like modeling task. As I mentioned that because we can do invariant modeling over different tasks, we can really use transfer learning to even speed it up by combining multiple data from historical data and build a transferable model to speed up the learning of the target domain. As a result, we can use machine learning to automatically generate code that gets to the state of art or even better than existing human-engineered effort on NVIDIA GPU, ARM CPU, and ARM GPU, and specialized accelerator. This is a system that's already used in multiple companies in product, and I'm very excited to tell about it. So please come to our poster 104 at the 5th floor today. Thank you very much.
Thank you to the Spotlight presenters. Now we have an oral, so it's James Whittington from University of Oxford talking about generalization of structural knowledge in the hippocampal entorhinal system.
So, hi there, many thanks for having me here. I'm going to tell you about generalization of structural knowledge in the hippocampal entorhinal system. The problem I'm going to try and tackle is how do we generalize structural information over sensory particularities. This allows one to quickly infer and understand new situations that share a common structure, and therefore reduces the burden on learning. For example, if we know the structure of news programs is that there's an important beginning, a worthwhile middle, and a funny end, then if we see a funny clip on the news, we can infer the program is about to end. We want to find a mechanism for learning and generalizing this sort of structures. Okay, let's have a look at a more concrete example. So here we have a maze which consists of a sensory object at each location. This can be separated into structural elements, i.e. the relationships between the objects, and the objects themselves. This is exactly going to be like our solution to this problem. We propose an explicit factorization of structural information away from sensory information. And further, we propose a mechanism for combining the two in a conjunctive code that represents the current environment. This explicit factorization of structure from sensory experience allows a new conjunctive code for any environment that shares the same statistics. For example, if we shuffled the sensory stimuli like so. This also neatly maps on to the hippocampal entorhinal system with grid cells in medial entorhinal cortex representing structure and cells in the lateral entorhinal cortex representing sensory stimuli. And finally, cells in hippocampus will represent this conjunctive code. We use spatial cognition as a starting point to tackle our generalization problem. This is so that we can use the well-characterized representations of space to both leverage and compare to the learned representations of our model. So I'm going to describe to you a few different types of cells now. You don't need to remember them in any way, but I just really want to highlight a few key points. The first one is how do we actually measure these cells? Well, there'll be an electrode recording a cell as an animal moves around in an environment. The black line there is the trajectory of the animal as it waves around, and the red dots every time this particular cell is active. This can then be changed into a rate map. The second point that I want to raise is that there are loads of different types of cells in the hippocampus and entorhinal cortex that care about space. For example, these Nobel Prize-winning grid cells, which fire on a regular grid, and the Nobel Prize-winning place cells, which fire in one particular location. There are also cells that care about distance and direction away from objects. For example, these object vector cells in entorhinal cortex, and these landmark cells in hippocampus. And then there are also cells that care about borders. So it looks like there's loads of different types of these cells that all seem to serve a disparate function. But I'm going to show you later that they can all be unified under a single common framework. The last point of this slide is I want to say that these entorhinal cells at the top, they generalize, whereas the hippocampal cells at the bottom do not. For example, those object vector cells responds the same to every of those white objects in the environment, whereas the landmark cells only respond to a subset of those. We're going to unpack that point now. The distinction that entorhinal cells generalize, whereas hippocampal cells do not, is true amongst all the cell types. A famous example is remapping. This is where two place cells that fire next to each other in one environment, for example, that yellow one and that green one, do not necessarily fire next to each other in a new environment. In fact, they appear to be almost totally relocated. This is not true for the entorhinal cortex. If we look at the grid cells at the bottom, two cells that fire next to each other in one environment also fire next to each other in the other. This is consistent with our proposal. These entorhinal cells encode generalizable statistical knowledge, whereas these hippocampus encodes environment-specific knowledge. Considering the wealth of experimental evidence, we have a chance to really understand this problem via neuroscience. We know a lot about the input to hippocampus, and so we're going to use arguments about generalization to formally understand what the entorhinal cortex is doing. Okay, to link space to generalization, we're going to consider a simple task. The task set up these 2D graphs with a sensory object at each location. This is going to be hidden to the agent, though. All the agent's going to see is a sequence of sensory observations and the associated passive action taken. The underlying hidden structure of the sequence will not be observed. This is going to be done in multiple different environments of different sizes and different arrangement of sensory stimuli. The agent is going to be tasked with predicting the next sensory observation. This is going to be often only possible if you understand the rules of space. For example, in this pink world, as the agent wanders around, it comes back to itself. How's it going to know that there's a motorbike there unless it knows things like a right followed by a down, left, and up means you're back in the same place? But also, how's it going to remember there's a motorbike there if it doesn't have a memory? Okay, so we're going to delve into that now. Similarly to how it's believed that the brain stores episodic memories, we're going to use Hebbian auto-associative learning of memories, i.e. fast waits between hippocampal cells, sort of using the sorts of equations shown there. Here, we saw the sensory representation of a motorbike, followed by the sensory representation of a book, and we can see that the weights are increased between coactive neurons. This here is a normal attractor network, where stored sensory codes are completed by partial sensory inputs. For example, if we present this half motorbike, it makes an initial partial guess at the memory, which then gets completed by attractor dynamics to retrieve the full memory. I'm going to show in a moment that if we use conjunctive memory representations, then we can retrieve memories based on positional codes alone. This is going to be very important if we want to make predictions. So making our memories a conjunction between the blue structural information there and sensory representations shown in red allow us to retrieve memories based on structure alone. This is because we can make our initial partial guess at the memory based just on the structural information. And this is what's going to allow us to do these sort of fun things like predicting sensory observations when we return to a previous location, like loop closure there. The key is going to be, how do we get these blue representations to be the same for both memory storage and memory retrieval? Okay, so we'll end up with memory storage via Hebbian learning and memory retrieval via an attractor network. Of course, the key remaining question is how on earth do we actually learn those blue structural representations such that they generalize? They're going to need to know the rules of space. As I said, we're tasking our agent with predicting the next set through observation, so it's going to need to learn things like left is the opposite of right, and going in a loop takes you back to the same place. If our blue representation can learn that, then it'll have the same representation when returning to a previous state, which then allows for the correct indexing of memories. We don't know what these blue representations are going to look like, but we're going to train them via backprop, and we'll discover that we see sort of entorhinal cell representations. Okay, so I'm going to present some high-level modeling details now. You're very welcome to switch off for a couple of moments, but there's just a couple of key points that I wanted. One of them is that we're mixing fast Hebbian learning of memories with slow statistical learning of structure. The previous slides can be represented in terms of a generative temporal model with memory. So this up top is our structural representation, along with the passive actions given. The most passive actions for the neuroscientists out there are our equivalent of head direction cells. This here is going to be our attractor network, which is going to be retrieving memories based on a structural input. We're going to optimize within the variational autoencoder framework, and so we need an inference network. This is going to be for inferring our conjunctive memories. So we're going to take our sensory representation, we're going to combine it with our structural information, and form a conjunctive memory representation. We're going to train this all by backpropagation through time. So backprop's got to learn how to use an attractor network for storing and retrieving long-term memories. And again, we're mixing fast Hebbian learning of memories with slow statistical learning of structure. Okay, well, let's have a look at the behavior of our network. We can see that our network can remember events after only ever seeing them once before. This is one-shot learning, and that's consistent with the hippocampus forming episodic memories. Our network is also able to predict sensory events, even if it's taken an entirely new direction to return to that state, i.e. these loop closures. This is only possible if you've learned generalizable statistics about the space. We also see that our network is able to remember memories for many hundreds of time steps. Even though backpropagation through time is truncated at 25 steps, this indicates efficient storage of memories. Okay, let's have a look at the sorts of structural representations that our network learns. We're going to have our network diffusing through the space of different sizes and distribution of objects like shown. We can see that our network chooses to represent relational information in a grid-like code periodic over space. And we can see that that representation generalizes to different environments of different size and distribution of objects. We also observe spatial hierarchies that is representing relational information at a different scale. And we also observe different phases. That is, it's like the same grid code but shifted along. These are all properties that biology displays too. And we also observe band cells, which are also observed in entorhinal cortex. Let's have a look at the representations learned in our attractor network. That's our equivalent of the hippocampus. We can see we get representations that look a lot like place cells, and in a new environment, they remap, again, to totally random relocations. And again, this is exactly like what's observed in the brain. Okay, so far, we've just had the agent diffusing around the environment. But of course, animal behavior is often not diffusive. Animals like to hang around near borders or head towards shiny objects. So let's see what happens, what our network learns if we get our agent to exhibit such behaviors. Making our agent spend a lot of time near boundaries makes our network learn to represent border cell-like representations, which generalize across environments. Getting our agent to head towards many shiny objects, we see the emergence of object vector cells. These are cells that respond at a certain distance and orientation from these red shiny objects. And we can see that they generalize both within and across environments. So we see that these entorhinal cells structural representations generalize. This is not true for the cells in our hippocampal layer, which we see the equivalent of landmark cells, which are like these object vector cells, but they don't generalize to all objects in the environment. And again, this is exactly like the brain. So these results suggest these entorhinal cells are optimally encoding transition statistics. Thus, we present the notion that there is a unifying framework linking together all these entorhinal cells, and that is representing basis functions for describing transition statistics, both diffusive and behavioral. So although the learned representations are in great similarity to those observed in the brain, our mechanism of generalization, that is the conjunction between sensory and structural information, relies on experimentally unknown phenomena. So we thought we'd test to see if we can find it in the data. So our framework predicts a preserved relationship between grid and place cells after remapping. So for example, a place cell which fires at the same location as a grid peak in one environment, when it remaps, it should also remap to a location that's consistent with that grid cell. That's exactly what we're going to look for. Does grid cell firing correlate across after remapping? And that's exactly what we find in the data, and we can see that it strongly survives permutation tests. And again, performing these analyses on our model, we see these same results. This is the first demonstration of non-random remapping in hippocampal data. This was predicted by our model. So to conclude, for machine learning, we've built a network where fast Hebbian learning interacts with slow statistical learning. And we've represented and generalized abstract structural knowledge. For neuroscience, we found an interpretation of place cells, grid cells, and remapping that offers a mechanistic understanding for the hippocampal involvement in generalization. We've also provided a framework that unifies many of the different entorhinal cell types as building basis functions for transition statistics. And we also suggested that spatial cognition is just one instance of a more general coding mechanism organizing knowledge across multiple domains. Many thanks.
Well, thank you, James, for a great talk. So there are microphones here. Step up to the microphones, and if you have questions. So we do have a little bit time for questions. So have you been thinking about how to move this beyond the 2D grid?
Yeah, so exactly the modeling. Yeah, the modeling approach that we've taken is totally agnostic to the rules of 2D space. So it's not just spatial, of course, but it generalizes to any 2D environment. But whether it be on a 2D grid itself, so haven't looked at that. Definitely a very interesting thing, and something we're definitely considering at the moment.
There's a question here.
So you seem to make the assumption that the Hebbian learning is doing the mapping with a new location that's going to be associated with a new object, right?
Yes.
So is this coherent in terms of time scale with the evidences of Hebbian learning that have been observed in the data, which usually happens over minutes or something like that?
So it's certainly known in the hippocampus. You get things like detonator synapses, which make immediate connections. So yeah, that is definitely consistent with that.
Yeah, hey. Hey. Great presentation, it was excellent. One really quick question and one slightly more complicated one. The quick one is, when you are doing the fast Hebbian learning, were you also training the Hebbian learning rates with backpropagation, or were they fixed?
So in this case, they're kept fixed, but in theory, you could also learn them as well.
Awesome. And the other one was, in this particular work, the actions were passive. In what directions do you think, what extensions do you think would require to take that to an active inference setting?
Yeah, I mean, it would be combining it with reinforcement learning, which is another game, and I chose not to do that this time. Exactly right.
Excellent, thank you.
Well, let's thank James again. We are back to spotlights. The next spotlight will be presented by Jeffrey Chan, once we're ready. The title of the spotlight is a likelihood-free inference framework for population genetic data using exchangeable neural networks.
All right. Hi, everyone. I'll tell you about a likelihood-free inference framework we developed for population genetics data using a neural network that we devised that's exchangeable. So briefly, population genetic data is usually organized in a binary matrix. So you can see that the rows are the individuals of DNAs that we sampled, and the columns are SNPs, or we can think of as sites that have mutation. And the ones are locations at which an individual at a certain position has a mutation, and 0 means they don't have a mutation. And so typically in population genetic data, something we might be interested in is trying to see what positions or what mutations correlate with the disease. So we might run some statistical test or some regression with the disease on the right with this matrix of mutations. However, one issue is the individuals are not sampled IID. There's some evolutionary generative model that governs how this data is created. So without accounting for the generative model, we might get a lot of false positive rates. So the generative model that we're interested in is the coalescent model. And so briefly what that is a binary tree governed by a series of Poisson point processes. And so you can see here the leaves indicate the individuals at a given position. So this is at one position in the DNA, these are what the individuals have. And so our generative model generally produces a series of correlated binary trees. So you can think about it as a billion-dimensional set of binary trees that are correlated. And with anything this combinatorial and this high-dimensional, we can't even evaluate the likelihoods point-wise. However, what we can do is we can quickly draw samples given parameters. So these parameters might look like they tune the correlation of neighboring trees, or they tune the length of the binary tree, or they tune how bushy the tree is. So we can draw samples data from this set of, so basically given a set of parameters, we can draw data from it. So this kind of allows us to use likelihood-free methods. So basically, we access the simulator. So traditional likelihood-free methods, such as ABC, use a simulator to produce data and see if it's close to the data that we've observed. And if it's close, then we just keep the parameter sample that we chose, and we'll eventually create a posterior this way. There are a lot of issues with this approach. One is it scales poorly because it's just some sort of high-dimensional nearest neighbor, and it's very difficult to tune. So our approach is that we devised a neural network that respects exchangeability, and it has a lot of properties that are useful in the population genetics setting. So first, in population genetics, the rows are exchangeable. So it doesn't matter in which order we have the individuals. So we need to encode permutation invariance within our neural network to learn the mapping from the data to the posterior. Second, in science, often we're interested in calibrated posteriors because when we make a scientific discovery, we need to know the amount of uncertainty around this posterior. And third, since we use amortized inference from the neural network, it's very natural that to diagnose and tune our hyperparameters. And last, this is generalizable to a bunch of population genetic settings. And come check out our poster tonight. Thank you.
Thank you. The next spotlight is presented by Cheng Zhang on generalizing tree probability estimation via Bayes nets.
Good afternoon, everyone. My name is Chen Zhang. I'm going to present generalizing tree probability estimation via Bayesian networks, which is a joint work with Frederick Mason. In molecular evolution, phylogenetic trees are used to model the evolutionary relationships among various biological species or other entities. And this idea of using trees for evolutionary modeling actually goes back to Charles Darwin and have really revolutionized our understanding of the evolution processes of the nature. And one of the core problems in molecular evolution is then to estimate the probability of those phylogenetic trees based on the collection of DNA, RNA, or protein sequences. And this is usually done via Markov-Chain-Monte Carlo, and the posteriors are typically estimated using those MC samples. However, the current approaches of using those samples are unsatisfactory. The common choice of using sample relative frequency does not generalize. So as a result, it usually requires a impractically large number of samples that takes extremely long time to collect. A previous effort that tried to mitigate this issue is to use conditional clade distribution, but it's not flexible enough for real data. So what is the best way to use those MC samples? Or in other words, if we're given a collection of trees, how can we estimate the probability of those trees appropriately, right? So in our paper, we proposed the subset Bayesian networks, which is a general probability estimation framework for phylogenetic trees based on MC samples that can generate strong sample trees, but also provide accurate approximation for those real data posteriors. And the key is to harness the similarity of trees properly. Just a few notation to set up the problem. Here's a rotated phylogenetic tree looks like, right? All the leaf nodes are associated with different labels that corresponds to different species, and we denote the leaf label set as X. We call it a non-empty subset of X a clade. On the tree, the clade actually corresponds to the leaf label set of the subtrees. Now, following the splitting process of the tree can be represented as a collection of non-trivial clade, which we call clade decomposition. And another use for local structure is called subsplit, which we define as an ordered bipartition of the clade. And examples are here: C2, C3 is a subsplit of C1, and C4, C5 is a subsplit of C2. And following the same splitting process, we can have the following subsplit decomposition. And actually, this set of subsplit or clade decomposition or representation of trees allows us to design distributions that can reflect the similarity of trees through those local structures. Okay? And actually, that inspires a natural Bayesian network formulation for tree probability estimation, which we call subsplit Bayesian networks. A subsplit Bayesian network, or SBN for short, is a Bayesian network where the nodes can take on subsplit or singleton clade values, and it contains a full and complete binary tree as a subnetwork. As shown in the right panel, each tree can be represented as a unique subsplit decomposition that can be represented as a compatible SBN assignments. We therefore can use the product of these conditional probabilities to estimate the probability of the trees. Actually, and this SBN formulation actually provides a valid probability distribution over the entire root tree space. Actually, we can adjust SBN to capture more complicated dependency structures among tree distributions. Okay, and learning SBN could be fairly straightforward. For root tree, we can use maximum likelihood. For unrooted trees, which is a common type of phylogenetic trees, we can view them as a root tree with missing roots. So we can marginalize the root node over all compatible root subsplit to obtain a valid probability distribution over the entire unrooted tree space as well. As a missing data problem, now we can use expectation maximization for learning, and we can also, in the paper, we'll provide an alternative method that is simpler but faster based on maximizing a heuristic lower bound. We can also incorporate regularization when necessary. Here are some results we obtained on a real data set with multimodal posture, where the conditional independence assumption made in a previous effort, CCD, is unlikely to hold. We see that SBN outperformed the other method, and especially in the weak data regime, and it also significantly reduced the bias introduced by CCD. And when applied to a wide range of real data sets, we see that SBN consistently outperformed the other method, and regularization is useful in most cases. And there will be more information in the poster session. So if you're interested, come to our poster number 123. Thank you.
Thank you. The next spotlight is presented by Ofer Lindenbaum on geometry-based data generation.
Hi, my name is Ofer Lindenbaum, and today I'm going to describe Sugar, which is a new method for data generation based on the geometry rather than the distribution. And this is a joint work with Jay Stanley, Guy Wolf, and Smitra Krishnaswamy. We're all from Yale University. Traditional generative models such as GANs and VEE typically try to generate points based on the existing distribution of samples. However, this does not take into account the geometry of the samples. It may lead to biases or loss of important transitions and rare populations. Instead, in Sugar, we present a new paradigm in generative models. Sugar first learned the geometry of the data and then aims to equalize the distribution of points along it. It does so by augmenting points specifically in the sparse regions of the original samples. To learn the geometry, we use diffusion-based manifold learning. By looking at local affinities, we impose a random walk on the existing samples. Then, by looking at different scales of this random walk, we can capture both local and global structures. To synthesize points, we first initialize samples in the original feature space. Then, by applying the diffusion process, we pull the points towards the manifold. However, a naive application of this process would pull the points towards the dense regions. To make sure that points are being pulled towards the sparse regions, we use a recently proposed kernel, a measure-based kernel. This kernel allows us to separate the geometry from the density of the points. Specifically, by using a sparsity measure, we upweight the probability of transitioning towards the sparse regions of the original samples. An interesting question which arises in this study is how to initialize these samples in the original space in order to end up with uniformly resampled points. In this proposition, we show how using careful perturbation and a specific choice of a generation level for each region, we can ensure such density equalization. As a first application of sugar, we look at imbalance classification. Here we applied sugar to 61 data sets with an imbalance ratio from 2 to 150. Sugar not only improves classification performance, but also outperform existing methods which are specifically designed for this task. Sugar can also be used for unsupervised learning. Here we've applied sugar to 115 data sets and show that clustering performance is improved by looking at the RAND index. Another major potential application of sugar is in high-dimensional biological experiments. In this example, we've looked at 4,000 single-cell RNA-seq measurements collected from a continuous process of early hematopoiesis. Sugar was able to recover this latent transition of B cells from early stage until maturation. Based on this transition, we were able to validate results by Veldon et al. Next, by looking at known biological markers, we were able to validate that the generated cells do follow expected biological properties. In this case, their canonical correlation has even increased by comparing to the original sparsely sampled points. I thank you guys for listening, and please come to our poster session for more details and more applications of sugar.
Thank you. The next spotlight will be presented by Anuj Sharma on point process-latent variable models of larval zebrafish behavior.
Thank you. As mentioned, I'm going to be talking about point process latent variable models of larval zebrafish behavior. So why are we talking about larval zebrafish behavior? The larval zebrafish is a key model organism for systems neuroscience research. Its brain is optically transparent, which lets us monitor the activity of all of its neurons simultaneously. While the tools we have for understanding the zebrafish brain are fairly comprehensive, our understanding of its behavior is relatively limited. So we've built a system that lets us capture high-resolution video of larval zebrafish behavior. Basically, we let the fish swim around in a large tank, feeding on paramecia, as we track it with an overhead camera. The system has allowed us to capture around 40 hours of video across 130 zebrafish. On the left, you're about to see a real recording from our dataset, and on the right is how we parameterize each frame for our models. You'll notice that we capture information about the angle of the zebrafish's eyes as well as the posture of its tail. You'll also notice that the zebrafish swims around with brief flicks of its tail. These are what we call bouts. As the fish enters a hunt, its eyes converge, letting it focus on the paramecia. The resolution of our video is high enough that we can actually observe the paramecia go through the zebrafish's mouth and into its stomach. With this sort of data, we're interested in answering 3 key questions. The first is simple. How should we characterize different types of swim bouts? Again, the bouts are these brief flicks of the tail, and we can also think of them as actions. Second, what dynamics govern how these swim bouts are sequenced together over time? And third, how are these dynamics modulated by different internal latent states, for instance, hunger? We develop a class of probabilistic models to help us answer these questions. Our models view larval zebrafish behavior as instantiations of a marked renewal process. This requires us to model 2 things. The first is the intervals between bouts, and the second is the characteristics of each bout. Each bout is represented by 10 frames of video, each of which has a corresponding set of eye angles and tail postures. Again, what we observe are these intervals as well as these high-dimensional marks consisting of eye angles and tail postures. So the first component of our model is a deep generative model of these high-dimensional marks. We assume that each bout reflects some low-dimensional latent embedding and that there's some highly nonlinear function that maps from the low-dimensional embedding space into the space of observed characteristics. To cluster bouts in the low-dimensional embedding space, we then introduce a discrete latent state, and to couple bout types, which we can think of the discrete states as different types of bouts, to couple these over time, we include a Markovian dependency between the discrete states. Finally, our latent continuous state lets us model variations in the transition probabilities between the discrete states and in the interval distributions over time. To perform inference in this model, we first marginalize out the discrete latent states. This gives us a fully connected, but importantly, a purely continuous generative model. We can then use this to perform amortized variational inference over the latent continuous states and over the embeddings. We do this with a bidirectional LSTM recognition network. Given a sequence of bouts and intervals, this network outputs an approximate posterior over the latent continuous states and over the embeddings. For a particular configuration of these variables, we can then reintroduce the discrete states and reason about their posterior using pretty standard HMM message passing routines. So with these models, we're able to begin answering our key questions. First, we find that larval zebrafish behavior is well characterized by 8 discrete types. We're able to make this determination on the basis of likelihood on a held-out set of fish. We then find that these discrete types follow an interpretable transition matrix, which suggests that fish transition between hunting and exploration, and that these transitions are modulated by what we call J-turns and hunt ends. Lastly, our latent continuous state lets us reason about variations and differences between fish that have been fed prior to entering the tank and fish that haven't. We find that fish that haven't been fed are more likely to transition to hunt-related bouts and less likely to end hunts. Moreover, fish that haven't been fed exhibit shorter intervals between bouts. This basically means that they, as we might expect, they swim more often. So the next step for our work is to include other sources of data. So environmental dependencies, for instance, the location and dynamics of the paramecia you saw in the second video, as well as whole brain neural activity dynamics, like what you see on the right here. This might look familiar from Michael Levin's talk. But we also think our models have potential applications to a variety of other domains. So if you're interested in the neuroscience applications I spoke about, or in extending these models to other applications, or if you just want to see more cool videos of fish swimming around, we look forward to seeing you at our poster, which is poster number 1 on this floor at 5 p.m.
Thank you to the Spotlight presenters. Next on, we have another talk by Sabi Shiv Kumar on probabilistic population code based on neural samples.
Hello. I'm going to present work on a joint project with my lab mates Richard Lang and Ankani Chattaraj with our PI Ralph Hafner. I would also like to acknowledge our funding sources. There's a lot of evidence to show that the brain performs probabilistic inference. Let me give you an example to demonstrate this. Imagine you're driving and you need to take a turn ahead. For this, you need to estimate the curve angle, which is about 45 degrees here. What about this case? The best estimate may still be 45 degrees, but the certainty in that estimate is lower. Accounting for this, most drivers would slow down and be cautious. The two situations above can be formalized in terms of probability distributions, where the black and red curves represent the distribution over curve angle. While they have the same mode, we see that they have different variances, and it is presumably this variance that leads us to slow down. More generally, we can say that the sensory input is noisy and ambiguous. Estimates made from this input, like the curve angle, are uncertain. And it is behaviorally useful to represent this uncertainty for optimal decision making. Therefore, the question arises, how does the brain represent probability distributions? One perspective on this question is what one might call a forward engineering approach, where we imagine how we might build a brain. We are interested in getting the probability of some latent variables, like the curve angle, given the noisy input, like the images. We could approximate this distribution by a parametric form, and the neuron responses could represent these parameters. In this case, the mean and variance of a Gaussian. We could also represent the distributions by drawing samples from it. Here, the high probability region contain more samples, and the regions with lower probability contain fewer samples. In this case, the neuron responses could represent these samples. Furthermore, inference in these models can be done using existing machine learning algorithms like variational inference and Markov chain Monte Carlo. However, neuroscientists are faced with a reverse engineering problem. Imagine we do a classic experiment where a subject looks at noisy gratings of different orientation. Now, we record activities from the neurons in the visual cortex, a part of the brain that processes visual input. Specifically, we record it from V1, which is known to be orientation selective, and we get a pattern of neural activity like this. Now, we vary the orientation, and for each such orientation, we get a different pattern of neural activity. Importantly, the activity is different each time, even for the same input. Looking at these patterns, we can ask questions like, do the responses of the neurons they represent the distribution over what variables? Do they represent the parameters or samples? Are they binary or are they continuous? Do they represent the probability directly, or do they represent the logarithm of the probability? Models on such hypotheses are found in literature, often as part of robust debates as to which one is a good model. To answer this question, let us first take a popular case of parametric codes, a linear probabilistic population code, and see what it predicts in the case of the oriented gratings. Under this hypothesis, neural responses represent the probability of orientation. Specifically, for a linear PPC, this distribution belongs to a linear exponential family where the firing rate are the natural parameters. Let me expand what this means graphically. Here, each neuron has a stimulus-dependent kernel, h of s, and the neural responses weight these kernels. Finally, this weighted combination is proportional to the logarithm of the probability over orientation in this case. This model is similar to the product of experts model used in machine learning. Coming back to the original picture, we see that the neuron responses seem stochastic. So one hypothesis is that they are generated from a sampling-based algorithm. Here we illustrate this idea using a linear sparse coding model. So what is this model? Imagine we try to represent the image, the tree here, by a weighted combination of its parts, like the trunk, the bark, etc. And here we basically take a bunch of features, which we call as A, and multiply with some latents. This actually gives us a generative model of the images, because we call these features known as projective fields, multiply them with some latents, and then add some pixel noise to get the image. In fact, Olshasen and Field showed that training such a model on natural images with a sparse prior on latents give us wavelet-like receptive fields which are observed empirically. So the goal of the brain here would be to compute the posterior probability of the latents given some image. Let x of t denote a sample drawn from this posterior distribution. Under the neural sampling hypothesis, the responses of the visual neurons basically represent these samples. This mapping becomes straightforward where the latents are binary. Imagine we show an image and get these spikes. Now here each spike represents a sample from the binary latent distribution and indicates the presence or absence of oriented wavelets or projective fields. Neural sampling and PPC clearly belong to different representation classes. This has led to a debate in the field whether the brain is better modeled as one or the other, implicitly assuming that these are mutually exclusive hypotheses. But is that really the case? In our work, we try to put these in the same picture. On one hand, we have sampling, where the neural responses encode the probability of the distribution. This can be viewed as Bayesian encoding. On the other hand, we have PPCs, where the probability over S can be decoded from the neuron responses. This can be viewed as Bayesian decoding. And having laid them out like this, we can ask, are the two theories talking about different representations in the same or different model? To answer that question, we attempt to link the two models in the following way. We assume a linear sparse coding based and a sampling based encoding. We derive responses from the model for gratings with different orientation. And we decode the orientation from the samples probabilistically and show that the result is a linear PPC. Let us try to simulate the oriented gratings experiment. The experimenter chooses an orientation, in this case 45 degrees, and the subject views a noisy grating. We can then view the spikes in response to this grating as follows. One thing we notice is that the neuron with the highest, with a matching projective field had the highest probability of spiking. Now imagine we were just observing the spikes. Given this spiking, can we make an estimate about the orientation of the shown grating? We can make this estimate by looking at the neuron which spiked the most, and then looking at the orientation of its corresponding wavelet, which seems about 135 degrees. And in this case, it turns out to be the right answer. Formally, the decoding question asks, how likely is each grating orientation given the neural activity? Here, the current set of samples. The Bayesian approach would be to use each sample from use a sample from each neuron and then weigh it by the orientation of the corresponding projective field to get the probability of the grating orientation. In this case, the neurons which spiked were about 90 and 135 degrees, so it peaks between those two values. In this case, it's between 135 and 180 degrees. And here, again, between 135 and 90 degrees. And since the samples are stochastically generated, each sample could have come from a range of orientation. Combining the distribution from more samples gives us a sharper estimate of the true orientation. And we see that it is peaked close to the correct value. In fact, there's a nice geometric intuition for obtaining the probability of the orientation from a sample. Consider a 2D pixel space just for visualization, and we look at the different gratings on the space. We see that the grating templates lie on a manifold in the space. Now, we project each sample to this space by multiplying it with its projective fields. The probability of the grating orientation is related to the distance between this projected image and the corresponding template. Coming back to the original picture, we conclude that each sample of R can be represented as a parameter over S. Each sample gave us an estimate of the peak, and collecting more samples gave us a sharper estimate of the posterior. So in essence, there are two distributions here. One is a sampling code, whereas the other is a parametric code. The sampling code represents samples from an explicit variable, the causes of the image, and whereas the parametric code represents the probability of an implicit variable, the orientation. But we still haven't characterized the form of the parametric code. To do so, it would require some lengthy math, which I'm skipping. The details can be found in the paper or the poster. The important part of the result is that the probability over S is in the form of an exponential of a stimulus-dependent kernel, h of S, weighted by the neural responses. This is the same form as a linear PPC. Hence, depending on the variable, we obtain two different classes of models. One is a sampling code, whereas the other is a parametric code. One is an explicit code over the causes of the input, and the other is an implicit code over the quantity of interest. For the binary latent case, the sampling model turns out to be a direct probability code, where the firing rate, the average number of spikes, is proportional to the probability of spiking, whereas for the PPC, it is proportional to the logarithm of the probability. The sampling model can be also thought as a Bayesian encoding model, and the parametric, the PPC can be thought of as a Bayesian decoding model. So to summarize, there's been a debate in the field between two classes of neural representation of probability, PPC and sampling. The two may not be mutually exclusive if they are over different variables. In fact, we showed that a sampling code for latent causes of a given image can be interpreted as a parametric code over the orientation. And this is true for any represented variable, not just orientation. Furthermore, in a general case, the simultaneous representation of different classes of probability distribution is possible in the same system. We can draw an analogy to machine learning, where we train a model on general input and then use this pre-trained model to get task-relevant quantities. This brings me to the end of my talk. Please check out our poster in the next session, number 26, and thank you.
Thank you. So again, there are two microphones here. We have some time for questions. So can you tell us something about the next steps?
So the next steps would be to see on what classes of models does this result generalize to, and under what criterion and what variables can be decoded from a model. So this, we are just looking at visual input, but this potentially is a general result which can be applied to different models of the brain, different parts of the brain.
Can you give us an idea of what a physiological verification of something like this would look like?
So that's what. So if you had, there are a lot of experiments in literature where people have recorded neurons from V1 and got these spikes. But just because of the internal model the experimenter has, whether it's they think that the neuron is representing the orientation, that turns out to be a PPC. And if the person thinks that it's actually a generative model of the image, it turns out to be a sampling code. So I guess the important question is, in already collected data, what is the model you're using of the visual cortex? And that will sort of give you different answers as to what is the code which is being represented.
So let's thank the speaker, Sabi. So we'll have, to the end of this session, another round of spotlights. And the first one will be presented by Rosemary Kay on sparse attentive backtracking, temporal credit assignment through reminding.
Hi, I'm Rosemary. I will be talking to you about sparse attentive backtracking, a new way to do credit assignment through time using a mechanism called reminding in recurrent neural networks. So credit assignment in the most general term means the correct division or attribution of blame or error to one's past events or actions that leads to the final outcome. Credit assignment in recurrent neural networks are usually done using backpropagation through time. Backpropagation through time requires a detailed replay of all of your past events, and then it assigns a self-credit to almost all your events in the past. This is usually not an issue if your training and test data looks significantly similar. But what if you want to generalize to sets that are not exactly the same as your training set, but shares underlying similarities? Humans are amazingly good at doing credit assignment through time, especially compared to recurrent neural networks. Imagine a scenario. You're driving on the road, you hear a loud popping sound. You think nothing of it. 20 minutes later, you're stopping by the side of the road, and then you realize that you have a flat tire. Instead of doing backpropagation through time, which is remembering everything that has happened in the past 1 hour since you were driving, your mind will immediately bring you back to the mental state of when you hear this loud popping sound. This is a mechanism called automatic reminding in cognitive science. And there has been cognitive and neuroscience evidence showing that this automatic reminding plays a useful computation role in terms of ongoing cognition. So here the question we're asking is, can this be used for doing credit assignment through time? What does this really mean when it comes to training recurrent neural networks? So this means that we don't want to do a detailed credit assignment through every single event in the past. Instead, we want to sparsely select a few events in the past and then do a local credit assignment around those events. But how do we sparsely select those credit time steps to assign credit to? We use a well-known mechanism in deep learning called attention. Attention assigns important weights to your time steps or events in the past. So in the forward path, your sparse attention mechanism looks at some of the times events in the past and then selects the top 5, in this case, the top 3 events that it thinks is the most relevant. It summarizes these and then use them for your forward computation. On the backward path, instead of doing backpropagation through time, you only do backpropagation through those sparse events locally. So we perform on-par to LSTM trained using full self-attention. We perform on-par to LSTM trained using full backprop. In some cases, we outperform the transformers network as well. But probably the most surprising result is generalization. So in this case, we take test set that are not exactly the same as our training set, but shares underlying similarities. So in this case, as you can see, we train on the copy task of length 100, and we perform significantly better even on sequence lengths of 5,000. So please come to our poster number 23. Thanks.
The next spotlight is presented by Shuang Li on learning temporal point processes via reinforcement learning.
So, hello everyone, this is Shuang Li from Georgia Tech. I'll give a brief description of our recent work, learning temporal point processes via reinforcement learning. So this is joint work with the students and professors from Georgia Tech, and Nandu from Google Brain, and Shuaixiao from Ant Financial. So modern complex systems such as healthcare systems, social medias are generating a large volume of discrete events all the time. Typical examples of event data are tweets, retweets, crime events, earthquakes, patient visits to hospitals, and financial transactions. It is our interest to learn the temporal patterns of this event data. While when we want to propose models to analyze the event data, we are facing two major difficulties. The first challenge is the occurrence time of this event data are irregular and random. So this is a major difference from the traditional time series data, where the measurements are evenly sampled. So we want to propose an elegant model that can deal with the event data in a continuous time fashion without time discretization. And the next challenge is how to capture the underlying complex dependent structures of the event data. So because the events are not independent with each other, the occurrence of historical events might influence the occurrence of new events, and the events, they are influencing each other. So how to propose a flexible model to capture the dependent structure is also a challenging problem. So point process model provides an elegant tool to model the event data in a continuous time fashion without discretizing the time horizon. It directly models the intensity function of the discrete event. The intensity function is defined as the average occurrence rate of the discrete events within a short interval by conditioning on the history. So by specifying the intensity function as different formula, we can actually capture different temporal patterns of the event data. For example, if we assume the intensity function as a constant, this means the discrete events are uniformly distributed over the time. We can also specify the intensity function as a deterministic function of the time, and this will capture the complicated shapes of the intensity function. But the discrete events in this setting are independent with each other. And we can also explicitly capture how the historical events influence the future by explicitly write the intensity function as a function of the history. A typical example is the Hawkes process, or called self-exciting process. It is commonly used in social media. So it captures the phenomenon that occurrence of events will boost the intensity of the new events, and dependency will decaying over the time by introducing the exponential kernel decaying function. So for the point process model, so far the most widely used learning method is based on the maximal likelihood method. So we have to go through two steps. The first step is to parameterize the intensity function as a parametric or non-parametric form. The next is to construct a likelihood function in terms of the data, we can learn the intensity function by maximizing the likelihood. This type of learning method suffers from the risk of model misspecification. Because the quality of the model directly depends on how the intensive function is parameterized. If in some settings we do not have enough sufficient prior knowledge, so if the intensive function is not well defined, even the optimal model obtained by maximal likelihood will suffer from the model misspecification. So our new idea is we really want the data to speak for themselves. We propose a reinforcement learning method to learn in learning the temporal dynamics of the point process model. The idea is quite simple and direct. We directly generate events from our generative model and compare the generated events with the observed events. We also introduce a discrepancy metric to quantify such a gap and get the generative model to improve its behavior by mimicking the behavior of the observations. So using the reinforcement learning framework, we relate the generating mechanism to the stochastic policy we want to learn. The state of the policy captures all the historical events, and the action is the next event time. And our policy is parameterized by LSTM to capture the long-range and non-complex capacity dependent structure. And we also learn our reward function to get the policy to mimic the behaviors of the observation. We use the inverse reinforcement learning framework to learn the optimal reward function. It is, in fact, a mini-max procedure and very time-consuming. So we choose the reward function from the reproducing kernel Hilbert space and obtain an analytical formula of the optimal reward function. It can be directly evaluated by the samples and the observations. So in our overall framework is a reinforcement learning problem with a known reward function. And also, we just using a very simple policy grading method and obtain promising results. We evaluate our proposed method to two state-of-the-art methods and three conventional parametric methods in terms of the data description and runtime. And our proposed model outperforms the alternatives under these two evaluation metrics. So we have a poster later, so if you have any questions, we can talk more later. Thank you.
The next spotlight is presented by Nazimi Tetbo on precision and recall for time series.
Thank you. Hi, everyone. I'm Nazimi Tetbo from Intel Labs. Today, I will tell you about a new approach for evaluating time series classification algorithms. This is joint work with my colleagues from Intel Labs and our collaborators from Brown University. Our work is motivated by time series anomaly detection. Anomalies are patterns that do not conform to expected behavior, while anomaly detection is the process of identifying such patterns. Anomalies occur in many diverse domains, from autonomous driving to medical diagnosis, and their reliable detection can be safety critical. In this work, we observed two key properties about time series anomalies. First, most complex anomalies manifest themselves over a period of time, which we refer to as range-based anomalies. And secondly, applications may have domain-specific preferences. For example, in certain domains, such as cancer detection, detecting the beginning of an anomaly can be important to respond to cancer early, whereas in other domains, such as robotic defense systems, end of an anomaly is more important to detect in order to avoid irreversible actions. Likewise, applications may have preferences for different kinds of classification errors, such as false positives and false negatives, and possibly others. Given this, a fundamental question is, how do we evaluate the accuracy of anomaly detectors? For point-based data, traditional precision and recall model works well in quantifying how many of the real anomalies were correctly identified with respect to the total number of predicted and real anomalies. On the other hand, for range-based data, there are much more complex situations that can arise, and the traditional model is not sufficient to capture all those complexities. As shown on this example on the right-hand side, real and predicted anomalies rarely perfectly match with each other. In fact, they oftentimes are detected in a partial manner and oftentimes in fragmented ways. Furthermore, there may be domain-specific criteria in addition to this, such as time bias. As a matter of fact, none of the existing accuracy models today can fully capture these needs. For example, the classical precision recall and the Numenta anomaly benchmark scoring model, they both apply to point-based data. The beta parameter in F-score for the classical model can make a trade-off between false positives and false negatives only, whereas in the Numenta model, there is support only for early time bias. There are other metrics, such as the ones from activity recognition domain, which provide better ways to express fine-grained events, but they don't provide other support for things like time bias. As a result, in this work, we extend the classical precision model from point-based data to range-based data, based on 3 design principles: expressivity to capture unique criteria specific to ranges over time series, such as time bias; flexibility in order to weigh those different criteria flexibly for different applications; and extensibility in order to provide ways to extend it for other domain-specific needs. In our paper, we provide examples for how to customize our model for different domains, from cancer detection to robotic defense to emergency braking and autonomous vehicles, for example, by setting the time bias from front end to back end to middle end. In our experiments, we show how we can express sensitivities that other models, such as the classical and the Numenta model, cannot, as well as we show how our model is effective in evaluating and comparing multiple detectors against each other. More details in the paper. As a result, this work provides a new way of evaluating range-based anomaly detection systems in a tunable way. And we hope that in the future, our model can guide design of new training algorithms for anomaly detection for time series, as well as other more general time series classification tasks. Please come to our poster session right after this presentation. And we have a bunch of pointers on our slides to code data and our paper. Thank you.
The final spotlight is presented by Felipe Tobar on Bayesian nonparametric spectral estimation.
Hi everyone, thanks for staying until the last talk of the session. When you see this slide, you might wonder, why am I seeing Jimi Hendrix, stars, a brain, a broken car, and a whale in the same slide? I use this collection of pictures to illustrate the interdisciplinarity of knowing how things oscillate. In astronomy, by looking at a light curve, which is the intensity of a star through time, we can understand what the processes, the chemical processes in the star are. By looking at the spectral representation of a recording in music, we can perform better compression techniques or music enhancement technique. We can even design equipment, musical equipment. In fMRI, we get observations in the spectral domain, and even in fault detection. If we identify a component that is oscillating in a strange way, that might be indicative of a fault, of a future fault. So how do we characterize all the vibration or oscillating patterns of things, right? Well, one way of doing this is to work with the spectral representation of our data. And we're going to consider time series in this little presentation just for simplicity. So the spectral representation can be understood by decomposing our data into periodic components, as we see in the figure, and then represent our data by the power of each of these components. So in practice, how do we compute this spectral representation? We do it by computing one type of spectra using the Fourier transform. And we can compute the Fourier transform of a signal in general, as it is depicted in the first figure. But in real-world cases, we don't have a functional form of our data. We only have noisy measurements and missing observations. So it is impossible to compute the Fourier transform analytically. So what one does in practice is what I have depicted in the figure number 3. We perform an interpolation of our data, hopefully within a family where we can compute the Fourier transform analytically. The problem with doing this is that we lose the uncertainty related to the missing observations. So what we propose to do is to perform a density, a probabilistic interpolation, and then apply the Fourier transform to this posterior distribution over our data. We have to be very careful, and the main challenge here is that we perform probabilistic interpolations in a space of distribution for which we can then do the push forward through the Fourier transform, and that results in an analytic form for the distribution over spectra. As you might guess, one family that allows that, because the Fourier transform is a linear operator, is the Gaussian process. So we have proposed a joint generative model for observations, latent signals, local versions of our data, and their spectrum. And by doing so, we have worked with local versions of our data that allow us to handle non-stationary data, and also allows theoretical existence of these objects. The main consequence of this is that the observations, the latent signal, and the spectrum of our data becomes a multi-output Gaussian process where all the operations of marginalization and conditioning can be performed analytically. This turns the classic spectral estimation problem into one of exact Bayesian inference, and we can compute all these moments in closed form. A few examples that I will show very briefly in this slide is one where we have incomplete data, and we use our method to compute the posterior power spectral density, which is the one on the left. This is a heart rate time series. And as the model plays a Gaussian process prior in the spectrum, then the power spectral density becomes a stochastic process with chi-square marginals. And the particular thing about this process is that the posterior mean is analytical. That allows for efficient optimization, thus allowing us to find periodicities via optimization, which is critical in many applications. Thanks very much, and see you in poster 114.
Let's thank all the speakers of the session. So now we'll have a poster session in rooms 210 and 517.